The use of microscopes for the study of bacteria, fungi, algae, viruses, microbiology is divided into different disciplines: space microbiology, air microbiology, water microbiology, industrial microbiology, medical microbiology, virology, recombinant DNA technology, genetic engineering, and the boom that has created the dolly sheep in the world today and that died of arthritis, biotechnology. Microbiology has its inception to the year 1746 when Anton von Leeuwenhoek observed animalcules in water. He was actually observing protozoa. He was Dutch. He belonged to Delft in Holland, and he was a cloth merchant and he was a drapist, and he worked with men's accessories of cloths. And in his spare time, he worked with instruments, played with lenses, glasses, and his first microscope consisted of the source of illumination being sunlight itself. And he tilted two glasses, one at an angle of 45 degrees Celsius, 45 degrees, sorry, and reflected light and then observed the animalcules. So from 1743, he began writing, uh, giving his correspondence that he found animalcules, whereas in the year 1746, microscope was discovered. But even before that, in 1665, the physicist Robert Hooke, he observed silver of the cork and he said he found some cells. So, with the discovery of microscopes and with Louis Pasteur contributing to the refuting of the theory of uh, abiogenesis or spontaneous generation, that means life originating from inanimate objects, microbiology has made landmarks of discoveries and inventions that has reshaped the history of medicine. We had coronavirus, the vaccine that was discovered for coronavirus did make corona a very important herd immunity developed uh, virus. So we find microbiology has its significance in modern times, and they determine the shape of human history. Also, microbiology deals with space microbiology, the use of SCP, single cell proteins, for outer shuttle explorations where astronauts intake SCPs as food material for their survival during extraterrestrial or out of Earth, out of space orbit journeys. So, with spontaneous generation being refuted, and there is no spontaneous generation, but there is only biogenesis, that life gives rise to life, there began a series of discoveries of by Edward Jenner, who worked with cowpox virus in milking milkmaids, and he said since they were already infected with cowpox virus and exposed to the pustules, they did not contract smallpox. So basically, the eradication of polio, the oral polio vaccine that is given to infants, the BCG, Bacillus calmette-guerin vaccine against tuberculosis or Mycobacterium tuberculosis, the causative agent of tuberculosis. So we have a series of discoveries of these vaccines that are absolutely mandatory for a safe, immunologically geared human physiology. So we have microbiology dealing with medical aspects as well as We have soil microbiology that deals with the biogeochemical cycle, the carbon cycle, the nitrogen cycle in the soil. We have microbiology developed or reshaped by scientists like Vinogradsky, who said streptomyces and actinomyces that produces streptomycin is of prime importance in soil microbiology. So, from the microbiology at a very nascent stage through virology and development of vaccines, we have also cloning of Dolly, the sheep, that revolutionized the history of beginning of biotechnology in the world. However, Dolly met with its fate. It died of arthritis, an autoimmune disorder. So there are controversies with the development of biotechnology, but microbiology is a basic fundamental subject for the development of biotechnology and other uh, life sciences discipline. Then we also deal with the study of bacteria and viruses using microscopes. We have the light field microscope, we have the fluorescence microscope, we have the phase contrast microscope, we have the dark field microscope. They all use light as a source of illumination, whereas we also have electron microscopes, like scanning electron microscopy, where electrons, high speed electrons bombarded through vacuum, is used to scan the specimen or to penetrate into the specimen. And the secondary electrons that are emitted from the specimen are trapped in detectors, amplified, and the fluorescent image or cathode ray tube screen is there to give you the image. So we have development of instruments. We have physical and chemical methods of controlling microorganisms. Sterilization. Sterilization is a complete removal, both vegetative